Evening, doctor. My name is Wang Mengnan. It is our honor to present the transmission electron microscopy to you. Please next one page, Zhixiang. Yeah, a TEM consists of three main parts: the electro-optical system, of the vacuum system, and the control system. This is a diagram of the electro-optical system. From top to bottom, we can see the electron gun, several licenses with different functions, the sample holder, and the screen. When we use it, the electron gun produces a beam of electrons that pass through this license in the vacuum and hits to the sample. And then, when it passes through the sample, it carries unique structure information of the sample. And then we can project it on the screen and observe the shape, structure, and other features of the sample after magnification. As we can see, it is very efficient efficient instrument, which is able to magnify the sample 15 million times. How does it prepare to a normal light microscopy? Let's see the next slide. First of all, the electron gun of the TEM generates and accelerates the electrons, generates faster moving electrons, and it uses electromagnetic license, which allows the electron better passage. The last thing is that it is very high resolution, often 215 times higher than a normal light microscopy, and it can magnify the sample 50 million times. Such is powerful property of TEM, make it widely employed in scientific education and industrial field. My part is over. My groupmate Priya will present the next part for you. Thank you.
Hello, Doctor. I'm Priyashini, and I'll be presenting about the working principle of transmission electron microscope. The beam of electrons from the electron gun is focused into a small, thin, coherent beam by the use of condenser lens. This beam is restricted by the condenser aperture, which excludes high-angle electrons. The beam then strikes the specimen, and parts of it are transmitted depending upon the thickness and electron transparency of the specimen. This transmitted portion is focused by the objective lens into an image of fluorescent screen. Optional objective apertures can be used to enhance the contrast by blocking out high-angle diffracted electrons. The image then passed down the column through the intermediate and projective lenses is enlarged all the way. The image strikes the fluorescent screen and light is generated, allowing the user to see the image. Moving on to next slide. Interaction of electron with specimen. As electrons bombarded onto the specimen, the electrons are either transmitted, absorbed, or scattered. The image shown here has darker and also lighter areas. The darker areas of the image represent those areas of a sample that fewer electrons transmitted through, whereas lighter areas represent areas of sample that more electrons were transmitted through. Next, I'll pass to my groupmate Faris.
Thank you, Priya. So I will be continuing the next slide regarding TEM. So now we know the working principle of transmission electron microscope. So the advantages of TEM, first one is it provides high resolution and magnification. So we can get high quality and detailed images from using TEM. Second, to provide information on elemental and compound structure. And thirdly, it can be used widely in scientific, educational, and industrial fields. So, however, there are drawbacks of TEM. First, extensive sample preparation. Sample need to be thin and transparent. Secondly, a time-consuming process will be used for TEM analysis. Third, projection limitation, because there is no depth of sensitivity in images, and only 2D images can be produced. And lastly, damages due to radiation. Ionizing radiation can damage the specimen due to high voltages being used in TEM, and it could reach as high as 400 kilovolt. So for the application of TEM, the first is the analysis of structure and texture. So we can determine the topological, morphological, compositional, and crystalline information of a sample. Second, in semiconductor analysis, to identify the flaws, fractures, and damages of the in-depth up to micro-sized objects. And lastly, biological or medical studies, where we can investigate parts of cells and molecules. So next, I'll be passing the next slide to Tong.
All right, thank you, Faris. So coming to question and answers, the first question is why high vacuum environment is needed in the TAM? So high vacuum is required to prevent the scattering of electron beam with the air molecule present. Scattering of electrons will lower the beam intensity and stability, hence resulting a low contrast and obscure the details in the image produced. Alright, second question. What is the requirement of TEM samples? For TEM samples, the samples must be cut into a very thin cross sections to allow the electrons to pass through, and any samples must be dehydrated to withstand the high vacuum of the microscope and embedded in a suitable resins to permit a very thin sections to be produced. So coming to third questions, why TEM has a higher magnification than a light microscope? This is because TEM uses a beam of electrons where it has a smaller de Broglie wavelength than a visible light that is used in a light microscope, which hence this allows for the examination of TEM to produce a finer details of specimen. Next, how electrons is produced in TEM. So actually, the electron is produced by a component called tungsten filament in the electron gun. Current is needed to apply to the filament to achieve the release of electrons. So next question, what are the major differences between TEM and SEM? This question is the answer is mentioned by one of my team member in the earlier. So never mind, I'll just explain it again. SEM creates the image in 3D, while TEM uses a transmitted electron. Hence, it creates only a 2D image. Besides, the magnification of SEM is about 2 million times, whereas TEM can magnify up to 50 million times. So this is our reference, and that's it. Thank you.